Okay, so we're going to make a start now. Big welcome to everybody who's attending our webinar today. It's great to see you all. My name is James. I'm one of the course advisors here at the University of Manchester, and I am responsible for our transformative oncology program. So in my role, I will take you from your interest in the program right through to the application stage, and if you get an offer, and guiding you through that process. So during this webinar, we'd like to really encourage everybody to post any questions they might have. Tell us where you're tuning in from today. Tell us what industry you're working in currently, and yeah, we can engage with your questions and provide you some answers and information. So I'm joined today by Dr. Susan Johnson, who is the course director of our transformative oncology program. And it's a really exciting program that started this year. So I'm going to give Suze an opportunity to introduce herself.
Thanks, James. Hello, everybody. It's great to have you join us today, and hello to those of you who are going to watch this back afterwards. It's a great opportunity for us to talk about the programme. As James said, this launched this year, so we are up and running. It's an online programme. It's very exciting. Transformative Oncology is a very progressive and medically applicable programme, which is available online, and it's available to study part-time. And this webinar really is about giving you the opportunity to see a little bit of insight into the programme itself and the motivations behind it. A little bit of background from me. So I'm the programme director for the entire programme. I'm also the unit lead for the first unit, which focuses on tumour biology and the heterogeneity of tumours. I have almost 20 years experience of translational research. And during that time, I researched different types of cancers using lots of different approaches, techniques and technologies. The two things I've always been particularly interested in when it comes to cancer, firstly, is the genetic transformation that happens from a normal cell to a tumour cell, and how those tumour cells can then go on to hijack their microenvironment and to continue to communicate with each other and evolve. And the second aspect is the implication of these biological processes and how they can have an impact on the clinical management of that cancer. So those are the two strands that really inspired me to continue research into cancer. I completed my PhD with supervisors, one of which was a basic scientist or a discovery scientist, and one who was a clinical pathologist. So this really challenged me, I suppose, as a scientist, to really look at how the biological, look for the biological explanations that would underpin any of those clinical observations that were made. I think it's really, really interesting how the two disciplines can both complement and contradict each other at times. But as we move towards approaching precision oncology and precision medicine, bringing those two strands together is even more important than ever. So that's a little bit of background from me, a little bit of background of my experience. Yeah, I'm looking forward to exploring the programme.
Thank you, Suze. Thank you for that. Also in this session, later on, we'll be joined by Professor Stephen Taylor, who is the head of the Division for Cancer Studies here at the University of Manchester. And so later on, we'll be talking about some of the synergies between science and medicine in the field of oncology. But first of all, we're going to talk a little bit more about the transformative oncology program here at Manchester. And also, if you have any questions about the program or about anything we talk about today, you can post them in the Q&A or in the chat function. And yeah, later on we'll be talking to Stephen Taylor. So I'll just stop my screen share now, and we'll start talking about the Transformative Oncology Program, Suze. So you mentioned that you are the program director for this. So what was your motivation behind starting this program?
Yeah, it's a good question. So, I mean, cancer is a global concern. In 2020, there were close to 10 million cancer-related deaths. But there's also a global acknowledgement that there are real gaps in the oncology workforce. This has obviously been exacerbated by the recent global pandemic. There's a lot of catch-up to do. Cancer is an extremely complex collection of different diseases, and that's a really important part to remember. And it's a field which is really rapidly evolving with advances in technology and understanding about, underlying genetic diversity, etc. It just felt like it was a really good time to bring all of our expertise that we have here in Manchester together in a way which was more accessible to the global campus.
Excellent. So you mentioned that it's the first cohort has already started this September. So how are they getting on? How's the course going?
Yeah, so our first cohort, our pioneers for the programme. So they've just completed Unit 1 and the feedback's good. They are finding that they're able to fit their studies in around their work commitments. And they've actually reported being both sort of inspired and challenged by the content. So I mentioned before that Unit 1 really focuses on tumour heterogeneity and why it's important that we understand that from both a biological and a therapeutic standpoint. And within the unit, I actually invite some of our clinical experts in and challenge them with some of the questions around why it's important to understand the biological sort of architecture of the tumour and the composition of the tumour and the different cell types, etc., and what impact that can have on the treatment, the success of the treatment, and also side effects, etc. And what's been really nice is we've had some feedback from one of our students who's a systemic anti-cancer therapy nurse currently, and they're studying part-time, and they've actually been able to take some of their learning from Unit 1 into their practice, where they've been talking to one of their patients and being able to describe in much greater detail around what their treatment is doing and how it's working and what potential side effects and why they might come about. So that's a really nice sort of example of how the learning that's incorporated into this program can directly impact on your working practices.
Yeah, and I think that application that you mentioned to their workplace is something that we might touch on later on with Stephen to sort of discuss about that crossover between lab and bedside, that crossover there. So that's really interesting that your students have already started to see the benefits from that on the course. That's great. So the program is an online platform. It's an online masters, or it can be done as a PG Cert and PG Dip as well. So what have you seen the advantages of that study method for this program?
Yeah, so the program's designed to be flexible, so it's really agile. The content is medically applicable, which was something that was really important, and we spent a lot of time talking to current people working in the field of oncology to really ask them exactly what sort of learning that they'd like to access, and it needed to be obviously inspirational, but because it's part-time, you can build up your working, whether you're looking to upskill or return to work, or whether you want to fit it in with your working world. And it's developed also to appeal to a global audience, so it's available regardless of your time zone. So although the units are scheduled to run, they run for 8 weeks at a time. They're scheduled within a block of time, but during those 8 weeks, there is flexibility for you to access that learning at a time which is convenient to you. So, for example, at the moment, we have students who are in the UK, we have students in Hong Kong, we have students in Kenya. So, time zones are different. But we've also managed to find a time where everybody can come together for a synchronous opportunity to meet as part of a tutorial as well. The other advantage of doing it as an online program means that all of the learning can really be enriched by sharing experiences with diverse peers across the globe. So Different students have different job roles, so therefore they have different perspectives. You know, the facilities and the treatments available to them in their home country or within the institute that they work it can vary, so that can influence decision making, etc. So, you know, there's an additional layer of really rich of learning that's available by doing it in this format.
Yeah, that's really interesting. I think you know, even with this webinar today, we've got people tuning in from all corners of the world. So that's really nice to see, and it's great that the course also has that diverse group of students, and you know, they'll have their different ideas on things, but oncology is such a universal area of study that, you know, there's such a good opportunity to collaborate there with people from around the world. So transformative oncology then. So why is now a good time to be learning more about this field?
So current statistics in the UK are that 1 in 2 will be touched by cancer in their lifetime. And there's a real need to push the excellence of cancer care. And in the 21st century, that really needs the intervention of multiple skills healthcare providers, from pathologists to oncologists to surgeons and nurses, but also to the basic and discovery scientists, nutritionists, pharmacists. You know, all of these different job roles have key roles really in transforming the outcomes for patients. So the programme is designed to reflect the changing needs of healthcare and be adaptable for different healthcare systems that are in place around the globe. We've said before, it's medically applicable education, but it's also brings in that innovative research. It pairs it with therapeutic strategies that we're using right here in Manchester, and all of those different approaches to really achieve effective personalised care for patients. And that is the way we're going to achieve the best cancer care we can for the 21st century. But I would add as well that the programme of learning, it's not just about gaining knowledge about cancer biology and treatment, it's also about developing skills to apply that knowledge to specific areas. So whichever area that you want to excel, you know, it really encourages intellectual curiosity, creative and critical thinking, so that you can incorporate the technology and innovation throughout the cancer pathway, whichever job role you have. So our learners will develop a whole range of transferable skills, which will really help to enable lifelong learning and equip them to lead and evolve with the changing needs of cancer care as we move forwards.
That's it's interesting you mentioned the different job roles which the course can apply to. So have you seen that with the current cohort of students this year? Is there a mixture of professions and industries there?
Yeah, absolutely. So we have people in the UK, we have people working within the NHS, but we also have people in private practice. We have nurses, we have radiologists, we have radiotherapists. We have people currently in leadership positions who want to understand better about screening programs or early detection programs. You know, some of these are our real strengths here in Manchester, and we can bring our expertise to sort of demonstrate some of the exemplars that we have been able to put together within Manchester. And you know, the learning from that can be taken forward into whatever their working practice is.
That's I think that's prompted a question actually from one of our viewers. So Sahar said that he is currently working as a toxicologist. So he's done a master's in toxicology at Birmingham University. And so he was asking about his suitability for this course. He's very interested in cancer and actually did his final project on cancer therapeutics. So would this be a suitable course for Sahar?
It certainly sounds like you've got the background knowledge and the education that we'd be looking for in applicants. So yeah, absolutely. Please do get in touch, and we can work with you on your application.
Yeah, and on that, in terms of getting in touch about your application, I will provide contact details and information at the end of our talk. That's great. So that takes us on to talking a little bit more about the University of Manchester then. So How is the University of Manchester a leader in this field of oncology?
Yeah, so I mean, the cancer landscape or the cancer research landscape in Manchester as a whole is really quite unique. The Manchester Cancer Research Centre has a strong vision for creating a future free of the burden of cancer, and it has a unique partnership with the University of Manchester, Cancer Research UK, and also the Christie Hospital Foundation Trust. And this really allows us to push the boundaries of our sort of collective understanding, if you like, and we do that in a really collaborative way. The University of Manchester is internationally recognised as a world-leading university, not just for its teaching, but also for its research. And cancer is one of the 5 beacons of excellence here. Cancer Research UK is one of the world's largest funders of cancer research. And the Christie Hospital is world leading as a specialist cancer treatment centre, and is the largest single site cancer centre in Europe. They treat something like 40,000 patients per year. So, you know, cancer research in Manchester really exists as a sort of a nexus where our research is interconnected, it's collaborative, and it's impactful. So it's possible to translate a finding in the lab into better, more effective treatments for patients. So yeah, the expertise are here, and within this programme we've got most of them involved in contributing to the content of this programme.
Yeah, I mean, you mentioned the Christie Hospital, which is so well known here in the UK and worldwide as well, and it's literally just down the road from the university. The connections that the university has with such an institute is must make Manchester such a great place to work and do research in this field.
Absolutely, and there's a real appetite to bring together, and this is what we'll be talking to Prof Taylor about. You know, there's a real appetite to bring both the clinicians and the scientists together to sort of really make sure that we're doing the research in the areas that need to be done, so that they can impact directly into the treatment and the treatment options and the patient care. And that's in partnership with the Christie Hospital.
Mm-hmm. Yeah, that's great. We've had another question come in from one of our viewers. So this applicant studied an applied psychology program, applied psychology masters, and is interested in this course. So possibly not as directly related to oncology, but I guess there is some overlap there with psychology. Would this be a suitable program for a student like this?
If so, the program is designed to appeal to, you know, allied healthcare professionals, anybody who wants to work within the field of oncology or cancer as a whole. And there are, as I said, there are so many different job roles, and they're all equally as important. There is an entire unit that focuses on patient care, but there's also, you know, it's around understanding that holistic, it's having that holistic understanding of what we mean by cancer treatment and cancer research. You know, it's looking at the tumour, it's looking at the patient, it's looking at the different branches of oncology, so understanding the intersection between clinical, molecular and also surgical oncology, and how those different disciplines interact. So we'll be setting the scene and helping all of our learners to understand how that they all work together. But then we also touch on things like screening programs, strategies for early detection, the science of radiotherapy, you know. So if you're interested in moving into the field of oncology, this program provides a really broad understanding of lots of different aspects of that. So if that's what you're interested in, absolutely come forward, and we can discuss your background and your qualifications, etc., with you and guide you appropriately.
Yeah, absolutely. And I guess talking about the you mentioned a few of the different areas that you would look at in this program. So what would you say are the key topics covered in the program?
Yeah, so we start with talking about really starting to understand the challenges of tumour biology. So this is around looking at tumour diversity, that's between different tumour types, but also within a tumour itself. So looking at, you know, the microenvironment, the impact of the position, the place where the tumor arises, the cell type, the architecture, all of that, and how all of those things can impact on detection and treatment options and success of treatment as well. In the second unit, we cover cancer as an individualized disease. Now, this is quite a novel unit to be offering on this type of program. It's really important. Right now, we are understanding just how essential it is to take interest and understand the underlying genetic diversity of each of our patients. If we want to move towards precision medicine, it's important to understand that how underlying genetics, health, lifestyle choices can all contribute to individual cancer cases. So it's really appreciating that both tumours and patients are individuals. The third is all around established and experimental therapeutics. So this is really outlining all of the present, but also the future cancer treatment strategies. So moving into immuno-oncology, for example. Again, moving towards the idea that every cancer is different, every patient is different, and therefore every treatment needs to be bespoke to that patient. And then in the fourth unit, I think I've mentioned already, but in the fourth unit, we really introduce how the different approaches are taken between the different branches of oncology. So surgical oncology, medical and clinical oncology. So whether it's chemotherapy, radiotherapy, surgical intervention, or more likely a combination of all of these things, how these different branches work together to treat an individual patient. And those are the first 4 units. So they would put together, they would complete the PG, the postgraduate certificate. That's 60 credits. If you wanted to move on then to the diploma, we start to specialise in some of the areas where our expertise in Manchester are absolutely world leading. So the science of radiotherapy. So really start to have that understanding of one of the cornerstones of cancer treatment, detection of early detection, how important it is to detect cancers earlier, how much more likely it is to detect and then treat that cancer successfully, and how effective screening and different imaging tools and technologies can really help with that early detection. Then we move into the principles of personalized medicine. So this is really understanding the steps that are required to realize the goal, which is around precision oncology. And then, as I mentioned before, the final unit in the PG DIP is around delivering the best cancer care. And this is all about establishing an effective and efficient clinical pathway to deliver optimal results. Not just clinical though, it also requires the integration of the scientists, the diagnostic tests, the research, etc. It all feeds in together. So those are the kind of topics that are covered.
Well, I think that final topic that you mentioned about both the scientists and the clinicians, contributing to the whole process is something we'll touch on in the second part of this webinar. So that's a really nice to see that the program touches on that as well. So you mentioned just then about the future of cancer treatments, and I think that a lot of people who might be interested in this course might be thinking about that kind of thing, about future developments in treatments. That's something that they're probably looking at getting into themselves. So that sounds like a very interesting module. So maybe you could provide some more detail about those future cancer treatments and how this program might touch on some of those.
Sure. So, I mean, in Manchester at the moment, so we've just we've got there's a researcher that worked on his project within Cancer Research UK and within the Division of Cancer Sciences, and as part of a spin-out company, they started to develop immunotherapy. So we've had our first patient just very recently, actually, who has received their own CAR T cells. So this is a process by which a patient will go in and their immune cells will be harnessed and reprogrammed and then sent back into the body to attack their own tumour. It's absolutely mind-blowing, and it's happening now. So the trials are happening. The treatment is starting to really show some fantastic results. So this is a lot of understanding around the immuno oncology over the last couple of years. Previously, particularly, for example, in brain tumours, it was assumed that there was no immune cells there. they weren't present in that area of the CNS. Whereas now, you know, with new technologies and new approaches and new understanding, we realize that actually, you know, harnessing the body's own defense mechanisms is actually a really, really powerful tool that we can use against tumors. And that, yeah, and that's happening right now. So we have the unit lead for this, Jamie Honeychurch, is a really top level researcher in this area. And yeah, our students this year will be interacting with him in the coming months. So yeah, it's an exciting time.
Yeah, that is fascinating stuff. Actually, that's I've never even thought about the idea of CNS not having these immune cells, or, you know, it's that's really cool, very cool actually.
We're learning all the time.
Yeah, I mean, it's such a fast moving area, isn't it? Like the work that goes into it is quite amazing, and it must be quite hard to keep on top of it, I would imagine. But I guess, you know, this sort of program at the university would give, you know, give students a really solid, you know, footing to move on to, you know, these innovative treatments and technology.
Absolutely, and that's the case if you're interested in it from a scientific point of view, from a research point of view, or indeed understanding, you know, how those treatments work if you're administering to patients. So yeah, I think there's a lot of different routes that this program can take you. You know, there's the sort of research route, there's the healthcare route, but there's also, you know, administrative route. If you want to go into science policy or marketing, science communication is a massive area. You know, people really want to know and understand about cancer, as I said before, particularly in the UK. 1 in 2 is the statistics we're looking at now. So everybody knows somebody, or if not themselves, then somebody close to them that's been affected by cancer. So, you know, there's a real appetite to understand. So things like medical research charities or funders, you know, the skills that you learn within this program are really transferable in that way. Of course, within research, there's academia, if you wanted to go on to do a PhD, pharmaceutical industry, biotechnology, or then the healthcare side. So whether you're patient facing, so in a hospital or a private clinic, or even in a community clinic, or whether you're in the sort of diagnostic services, or, you know, data analyst, or, you know, a sort of center manager, having this broader and holistic understanding of cancer across the board is really invaluable to help shape those sort of different workplaces.
Absolutely. What I'll do is I'll just share my screen and give our viewers a little bit more information about the oncology, the transformative oncology program that we have, and then also welcome you all to ask any questions if you have any questions about the program, about how to apply, or
I think you might have just muted yourself accidentally.
Oh, I did. Thank you.
We've all done that.
I was just mentioning about how this program is one of our online programs. So it can be done over 12 to 36 months, depending on which option you are on. So Sue's mentioned earlier about options to do it as a PG Cert, PG Dip, or as a master's qualification. And so on average, you'll be looking at about 15 to 20 hours of work study time per week. So it's something that, you know, most of our students fit around their employment. It keeps things, you know, flexible, and you can do it. Most of the content can be covered in your own time with the recorded content. And so our next enrolment for this program is in September 23. And so applications are open now. You can apply for the program. And I'll just put up my contact details here. So you can visit our webpage, our program webpage for transformative oncology. So if you just search for manchester.ac.uk/transformative oncology. Or if you just Google that, you'll be able to find that. And there's lots of information about the programme. And also you can email me at studyonline@manchester.ac.uk, and I will be able to answer any questions you have there. And we've been joined by Stephen Taylor now, who is Head of Cancer Studies here at the University of Manchester. So it's a great opportunity to have a discussion about our webinar topic today, which is the synergies between science and medicine. So, Suze, would you be able to just give us a quick introduction about this topic?
Absolutely. Hi to Stephen Taylor. Nice to see you. Thank you for joining us. Just to continue what we were talking about before, you know, and really for context, transformative oncology is about achieving better outcomes for patients. And it's looking for those opportunities for collaborative working, innovation and translational research, which our guest speaker here today is an absolute world leader in. So in this second part, we're going to discuss a little bit more about the interface between science and medicine, and why it's really important to succeed in this interface to achieve precision oncology. You know, discovery science and clinical medicine really need to work synergistically together. And the model for translational research traditionally is bench to bedside, but we also increasingly realize that we need to work from bedside to bench and then back again. So really, for the people on the call, the people who are interested in this webinar, if you're medically trained and you're interested in pursuing a career as a clinical academic conducting research in oncology, you will benefit from the understanding the underlying biology of a tumour and some of the basic science methodologies that would help you then to go on to plan projects or write grant proposals, and indeed to conduct investigations. And equally, if you're intending to pursue sort of translational research or develop maybe diagnostic platforms in oncology, it's hugely enriching to understand and appreciate the potential clinical application or impact of your discoveries and the experiments and investigations that you carry out in the lab. So on that point, this feeds very directly into Stephen's research expertise. On that point, Prof. Taylor, would you like to introduce yourself to the audience, please?
Yes, good afternoon, everybody. Thanks very much for the invitation and the opportunity to tell you a little bit about what we're doing as an exemplar of the Manchester way of doing things. So yeah, a bit of background about myself. I've been working started the early part of my career really working on understanding the basic cell biology, the basic cell biological mechanisms that underpin cell division, the cell division cycle, chromosome segregation, in particular that phase in mitosis when the chromosomes segregate, and understanding how that works correctly. And of course, this is important from an oncology perspective, because we know that tumor cells make mistakes during cell division. And we also know that a lot of drugs which are used routinely as chemotherapy agents will also target cell division cycle pathways, chromosome segregation pathways, and so on. So yeah, as mentioned, I spent 10, 15 years really working on the basic cell biology, understanding how all this works at a molecular level. And then what I really wanted to then do was then start to translate some of those findings into a much more clinically relevant context. So at that point, I moved my entire team from main campus in the university down to the Christie Hospital campus, and we now have a lab working directly over the road from the hospital. And what we decided to do was focus on ovarian cancer, because ovarian cancer is one of the most chromosomally unstable diseases. It's a disease where the antimitotic chemotherapy agent Taxol is used very extensively. And so ovarian cancer seemed like a very important disease for us to apply our very specific expertise. And what we did is we set up a reverse translational pipeline. So Sue's just mentioned bench to bedside. So that, I guess, we would think about as the traditional translational route where we take discoveries from the lab and we try and translate those into patient benefit. But again, as Sue's also mentioned, the bedside to bench approach, that reverse translational thing, is where we take samples from patients and bring those back into the lab to study them. So why is this so important? The problem with it, one of the major challenges with oncology is a lot of our model systems that we use in labs, what we've discovered over the last 10 years, they don't really reflect the complexity and the heterogeneity of the disease that we actually see in the clinic. So we have a very limited number of cell lines, of mouse models, and so on. And I think that's been a real limitation to making advances in our understanding. So when I moved my lab down to the Christie campus, we set up collaborations with clinicians, with the biobank staff. And now when women are being treated for ovarian cancer, we collect biopsies from them. They come from the Christie Hospital straight over the road to my lab, where we generate ex vivo cultures from these biopsies, and we're building a living biobank. So what do I mean by that? Is the traditional biobanking is you take a biopsy from a patient and you plonk it into formalin to fix it so that you can do the nice histopathology, you can make the nice sections and stain them with antibodies, and you can do the genome sequencing and so on. But what you've got is preserved tissue. You've got dead tissue. What we do is we keep the biopsy alive and we pull out the tumor cells, and we've figured out the cell culture conditions that allows us to maintain those viable cell cultures in the lab. So this is really exciting because we can do all the genomics, we can do the gene expression profiling, we can do the genome sequencing, but because we've got viable cell cultures, we can now do the mechanistic studies, and importantly, we can do chemotherapy experiments in the lab, and we can screen our living biobank samples with drugs to then understand the molecular determinants of drug sensitivity and drug resistance. So this is incredibly exciting. So this big collaboration between scientists, cell biologists, clinicians, biobank staff, we've now built a living biobank. We've got over 120 samples from about 80 different patients. This is the biggest resource of its kind in the world. And I think this is only possible because we've got this beautiful collaboration between one of the biggest cancer hospitals in Europe and one of the biggest universities in Europe. And it's that interface between the patient care, patient treatment, and academic research that has allowed us to do this. And it's incredibly exciting. We've got a number of projects now where we're using our living biobank samples to understand more about the basic biology of the disease. We've got projects looking at specific therapeutic modalities to see if we can identify therapeutic windows where some of our ovarian cancer samples will be sensitive to particular drugs. And what's now really exciting is we're now going from our reverse translational pipeline to making discoveries. We're now going back the other way. We've identified a very specific drug combination, a low-dose drug combination that we now want to, this paper's not been published yet, we'll be published very soon, it's going to come out in NAR Cancer, where this combination at low dose seems to be very effective against 90% of the ovarian cancer biopsies we've tested. So now what we want to do is, right, how do we now translate this finding back into the clinic to start thinking about those early phase 1 clinical trials testing this particular combination? We've got a little bit of work to do in the meantime. We can't just go straight from our living biobank straight into patients. We need to start doing some animal experiments in the meantime. We need to do some toxicity testing. We need to do all of that. I can't do that. That's not my area of expertise. But guess what? I know somebody who can. And so what we're doing is, again, building the teams, building the collaborations. We now want to get people who can take our living biobank samples, develop those animal models that will then allow us to do those drug combinations in an in vivo setting, to get that in vivo data, to get the toxicity data, and understand a little bit more about how it works in a more complex model, which will then inform the clinicians how they can then perhaps do those similar drug combinations in patients. And that's going to require collaborating with clinical trialists. It's going to require collaboration with pharmaceutical industry. There's going to be all the IP and the legislation and all of the regulatory stuff around that. No one individual can do all of that. I'm not an expert in all of these things, but I know people who are, and it's my job to pull those people together to create those teams that take these ideas from patient samples back to the lab and then back into the clinic. And I think that's something really unique about what we can do here in Manchester. And I think the training courses and the transformative oncology programs are things we're trying to set up will really give you a flavor of how this backwards and forwards from the clinic to the lab and back again, how that will really work in practice, such that you'll then be in a position in the future to be involved in projects like that. I think I shall stop there.
That's fascinating stuff. Thank you for that, Stephen. That's that's exactly what we're focusing this webinar on today, is that the topics that you've touched on there. So I guess a question to both of you then. So Stephen, you mentioned about this desk to bedside kind of crossover there. So why is it important to link clinical observation with scientific exploration to better understand tumor biology?
That's excellent question. I think it goes back to this, what we've really appreciated in the last 10 years is the incredible diversity and the heterogeneity of tumors. If we take ovarian cancer, for example, all patients that get ovarian cancer will probably go onto a very similar chemotherapy treatment regime. But of course, we now know that there are 5 different subtypes of ovarian cancer. There's high-grade serous ovarian cancer, there's low-grade serous ovarian cancer, there's mucinous, clear cell, and endometrioid. And these are driven by very fundamentally different mechanisms. So therefore, why would we expect patients with these different diseases to respond in the same way to the chemotherapy? So the way that the most prevalent form is high-grade serous ovarian cancer, and the treatment regimes have been developed largely based on high-grade, but they're given to all other 4. So that's one problem. Secondly, what we know, even within high-grade, it is such a diverse spectrum of disease, because we know now, and we didn't know this 10 years ago, that it's incredibly chromosomally unstable. These cells are constantly making mistakes. So when we look at different patient biopsies, they're just so different from each other. But then when we look at the cells just from one patient, they're so different to each other. There's so much heterogeneity within patients and between patients. And you just simply cannot capture that complexity by looking at the traditional models, established cell lines. There are about 40 ovarian cancer cell lines as part of the Cancer Cell Line Encyclopedia. There's maybe 20 of those are high grade. Some are good models, some are not good models. We've now got 120 from our patient biopsies. We've got more models than has previously existed that have been collected over the last 60 years. We've got a large array of models, and we're now seeing really for the first time just how heterogeneous and complex these diseases are. And so once it's quite overwhelming actually. What do you do with all of this complexity? One thing you can't do is ignore it. So of course, now that we've seen this complexity, we've exposed this complexity by doing the high quality sort of discovery science from our samples, now we can start to see the scale of the challenge we've got ahead of us, which will then certainly influence the way we think about taking our discoveries back into the lab. It sounds a bit all doom and gloom, but of course it's not. There are massive success stories. We know, for example, PARP inhibitors. These were discovered. It was discovered about getting on for 15 years ago that these drugs were synthetic lethal with cells that have BRCA mutations. That's now leading massive advances for women with BRCA deficient ovarian cancer. And then the question is, what do we do about the rest? And the more we understand about the biology, the more we understand about the complexity, the better positioned we'll be to come up with therapeutic strategies that are more likely to work.
I would actually add to that as well. I think it's a really unique time at the moment because technology, the advances in technology, particularly in the last 10 years, enable us to look even at the single cell level in a tumour. And that gives us an incredibly deep and, again, more complex understanding of that individual cell. And there are exemplars, obviously, from Manchester as well, where, you know, looking at the genomic screening through whole genome sequencing of individual tumours, etc., things like lung cancer, picking up specific mutations, perhaps in EGFR, for example, and then taking that through to the clinic and being able to pick out specific treatments that may or may not work on that patient, depending on whether that mutation is present or not. So screening and the capability of screening in individual labs, as opposed to, how long ago was the Human Genome Project? 20 years ago, where it took hangars of machinery all around the world as a real global sort of project working together. And I mean, I've seen a video of somebody walking alongside the printed out sequence of one single human genome, and it took months. Now in the lab, in a single sequencer, you can churn that out in a couple of hours. You know, it's more affordable, it's more accessible. So now we can get that data, as you said. Now we can get that information. It's like, how do we use that information? How do we select for patients that are, you know, going to benefit from specific treatments, and how do we match together that science and that clinical observation? It's an exciting time.
Yeah.
And no, just about one thing I'm very excited about at the moment. So we've got a living biobank. We're characterizing all of those samples. This is funded by Cancer Research UK, and they're funding us to do the gene expression analyses, the phenotypic analyses. But we've recently been awarded a really nice grant from the Medical Research Council, where we'll be able to do the whole genome sequencing on all of those samples. So, like Sue says, we'll be doing whole genome sequencing on up to 100 samples, and on top of that, for subsets, because it's quite expensive, we'll be doing single cell transcriptomics as well, to follow how these cells change as they become drug resistant. So absolutely, we've got this amazing living biobank, but then superimposing on top of that state-of-the-art technology platforms like whole genome sequencing, like single cell methods. Yeah, it's really very, very, very exciting. Costs money, so that's why we need to get grants, but we can do it, and it is very exciting.
Well, it's interesting you mentioned the advances in this technology. So now looking at time scale of things, how can this sort of technology development, how long is it from scientific discovery to being able to influence clinical management?
Yeah. So there's two parts to that, in the sense that, so an example I mentioned a minute ago was the PARP inhibitors. So those two papers that came out back to back in 2005, and they were just stunning papers, but they were based on, it was real discovery science, it was lab-based experiments. And 2005, 2015, by 2020, so 10 to 15 years, we were then starting to see really exciting results in the clinical trials. So you could argue that's quite a pretty rapid time frame, 10 years going from discovery to phase 3 clinical trials showing impressive clinical efficacy. So you'd hope that things would accelerate. But the trouble is, at the same time, what that is an example of is the low-hanging fruit. Okay, it took it took a lot of work and a lot of discovery and a lot of hard effort, but nevertheless, there's that beautiful biological relationship between inhibiting PARP and a BRCA mutation, and that gives you that synthetic lethality combination. We've now got lots of other really nice drugs inhibiting other things which are effective in some cases, but the real challenge is the coming up with the predictive biomarkers that will predict which of your patients are going to be sensitive or resistant, or their tumors are going to be sensitive or resistant to any given drug. So we've got PARP inhibitors and BRCA, so we know that works works really well. We've got the drug, we've got the biomarker. We've got lots of other drugs against many other cell cycle regulators, DNA damage checkpoint proteins, ATR inhibitors, CHEK inhibitors, WEE inhibitors, and so on. These are great drugs, but we haven't yet figured out how you identify the patient subpopulations that are going to benefit from those drugs. And until you do that, there's really no point taking those drugs into the clinic, because if you just go into a random population, the trial's likely to be negative. It's really about sort of salami slicing your patient cohorts into to identify the a priori which groups are going to benefit. And that can take, it can be a breakthrough that gives you the answer. It could take 10 years to work that out. And so I think while on the one hand we think things should always be accelerating, again, maybe we've solved some of the easier problems, and actually what we've got left are some of the more complex problems, which will, things won't necessarily accelerate. So again, to answer the question, it really is a case by case, depending on the complexity of the disease, the actionable drivers, and so on. And finding those biomarkers, that's crucial.
Yeah, absolutely. And I think you've just beautifully, without realizing it, you've just beautifully summed up the purpose of Unit 1 of Transformative Oncology. So for those of you who are watching who perhaps don't have this in-depth scientific background, you're perhaps more clinical, the unit really covers this holistic understanding. It really kind of touches on using exemplars, using world leading and real world examples, touches on the reasons why we need to understand this. You're not necessarily expected to understand in the depth that Prof. Taylor is able to describe it today, but having that perspective and having that understanding and being able to bring that into your clinical practice or in your scientific practice is really beneficial to really frame the work that you're doing in your day-to-day life. And actually something that we encourage within the assessment side of stuff is building a reflective portfolio. So these little light bulb moments that you learn as you go through the program, we encourage and we enable you to build a portfolio of that, which will serve you for your lifelong learning as you go through the program and beyond.
That's great. Yeah, I think that is a really nice thing about this Transformative Oncology program that you mentioned there. So I think that some of the viewers today might be interested in the field of research, and so talking about getting a research idea into the process, finding funding for that. So if our researchers were working in collaboration with clinical colleagues following a clinical observation, how difficult is it to formulate a research question which will attract funding?
Prof. Taylor's the expert.
Yes. Yeah, no, it's a very good question. So I think very often clinicians and scientists tend to think about these formulating questions in slightly different ways. And that can sometimes result in a little bit of confusion, people not necessarily talking at the same level. And I think one of the things I've learned collaborating with clinicians, and now I've now more recently had clinical trainees in my lab, is it does take some time to get those conversations to a point where you're really able to talk to each other and frame the questions. So you can come at it from a clinical point of view. What's the clinical challenge? So, in ovarian cancer, as I mentioned, we've got great, PARP inhibitors are fantastic. They work for a subset of patients, but the clinical challenge now is what about the rest of the patients that aren't going to benefit from PARP inhibitors? So that's a very clear clinical unmet need. What are we going to do about ovarian cancer patients that have got a particular type of disease? But yeah, how you then frame that into a research question is, where do you start? That's really very difficult. And so I think that's where you then need, as an individual, to take some, you need to take a step back from that sort of clinical problem and ask, okay, well, what do I know? What do we know collectively? What are the knowns and the unknowns? And maybe we need to, understand the disease better before we can start actually coming up with therapeutic strategies. And so then, okay, so what are the research questions that then come from that? And I think that will also take a collaborative process. Some people, they can have a light bulb moment and they can come up with questions and it takes a bit of creativity, a bit of imagination. It takes time and space to think about the problem. And sometimes you may not be able to formulate those questions in a sort of hypothesis-driven way initially. And you might need to just go back to basics and do some descriptive biology, some understanding. Let's do some genome sequencing. Let's analyze these different tumors. What do I learn from that? To see if that can then generate hypotheses that then can allow you to start formulating questions. So again, it can be quite difficult. It's difficult to answer that question in a very sort of abstract way without necessarily going into specifics. But I think the process that one uses to do it is to read, think, talk to people, read, think, talk to people some more, talk to some different people, get ideas, and then eventually, at some point, you've got to go do some research, whether that's experiments in the lab or whether that's meta-analysis of data that's already out there, and then see if you can then start formulating and crystallizing that process into questions and ideas. It doesn't happen overnight. I've been doing this for 20 years, and each idea and question is building on stuff we've done in the past. So yeah, not an easy question to answer, but an interesting question.
But it does give me an opportunity as well just to talk a little bit about the research element of transformative oncology. So if you choose to go to complete the entire master's, you can complete a research project in a lab-based project or in a clinical practice, or if you want to continue your journey online, there's a research skills in practice module, and that really covers some of the aspects that Prof. Taylor was just talking about. So ways to formulate that research question, how to go about doing a critical appraisal of the literature, how to design a study and the methodologies involved. But it also covers things like responsibilities as a researcher, so maybe something about ethics, communicating your research, so that's being able to communicate your ideas to somebody to sort of bring people around to your way of thinking, and setting up those networks. But also, again, as we've said, reflective practice, making notes of exactly what you've been thinking about and linking those with your thoughts, and then go back and do some more research, etc. So those skills are something which are specifically taught within the research skills and practice element.
Great, thank you for that. Thank you both for all those great insights into this field. It's been a fascinating talk, and appreciate your time and your efforts in sharing all that for our viewers today. That pretty much brings us to the end of our talk today. We do have actually just one final question from one of our viewers, Susan, who is a semi-retired nurse consultant with a background in oncology and hematology. She was a lead cancer nurse and also a palliative care nurse there as well. And she's an MSc in nursing. And she's asking, do I need to be working in the field to apply for this program?
No, absolutely not. If you're interested in upskilling, if you're interested in changing direction of your practice, or if you want to access this part-time purely for your own education and inspiration, then absolutely you're eligible to apply. And it sounds like, from what James has just said, it sounds like the background that you have would be really useful. So please get in touch and we can explore your application. James, I don't know if you're aware, but there's a question in the chat as well that Hazel's popped in. I don't know if that's where it's quite.
Oh yeah, I think that's right. So this is from Afsana on LinkedIn. So as a former cancer treatment, yeah. So she's mentioned about lots of different technologies that are involved in cancer treatment right now, and the wonderful work that, you know, all these practitioners, pharmacologists, biologists, genomics, and all these different areas. I mean, it is a real collaborative field, isn't it? And I think that's probably one of the nice things with the transformative oncology program, is that you get that collaborative feel, and people of different areas and different academic backgrounds. So that's something that is probably highlighted in your course and the cohort that you're working with now. Is that right, Suze?
Absolutely. And I really just wanted to draw attention to that, because the patient's voice and interacting with patients and understanding things from their perspective is really, really important. And it underpins what we do as scientists, it underpins what the clinicians do. So it's really wonderful to have that feedback from Afsana today. So thank you for taking the time to pop that into the chat there. It's really, really, it gives everybody who works in this field a real perspective to understand and remember that these are human beings that are, you know, undergoing some of these things. And sometimes it can be quite abstract when you're there in the bench with your lab coat on and a pipette in your hand. So, you know, just having that additional perspective, it really, really helps. So thank you for that.
Thank you, Suze. Yeah, I've just launched a quick poll for all our viewers. There's just a couple of questions, really quick yes or no questions that you can answer for us at the end here. We'll appreciate your contributions to that. And yeah, we'll finish there for today. But yeah, again, thank you very much to Suze and Stephen for those really insightful comments there about this field and the program that we offer here at Manchester. So again, if you have any more questions, you can reach me at studyonline@manchester.ac.uk. Also, you can visit our program webpage. But yeah, thank you very much for your time, everyone. And we look forward to hearing from you soon.
Thanks ever so much. Take care now.
Bye.